Hello everyone. Today I am going to talk about the very important topic that is nutrigenomics. It is one of the trending topic and it is a part of food science and nutrition. So first of all, let's know the meaning of the nutrigenomics. In this, two things is responsible: diet and gene. Diet, from which we can the nutrients can be obtained, and the gene. So the nutrients interaction with gene is called nutrigenomics. It is one of the basic and simple definition of nutrigenomics. The food that we eat, which contain nutrients like vitamins, that are fat solubles or water solubles. Fat soluble vitamins are A, D, E, K, and the water solubles are B and C. Minerals, carbohydrates, proteins that interact with gene, and then what are the reactions occur in our body that we have to study and which is known as nutrigenomics. So these are the contents that is mentioned in the upcoming slides, which includes introductions, interaction of nutrients and gene expressions, that is nutrigenomics, advantage, disadvantage, applications, future trend, and conclusions. So what is nutrigenomics is the nutritional genomics is the latest scientific discipline that uses modern genomics technology to study the relationship between genes, nutrition, and health. It explores the effects of nutrients on the entire genetic makeup, that is genome, proteome, and metabolome. Simply, nutrigenomics defines how the diet acts on the genes and changes gene expression, which is commonly prominent in cancer like non communicable diseases. Personalized nutrition connects this two-way relationship between nutrients and genes. This emphasizes how the consumed nutrients can affect our gene expressions as well as how our genes can influence response to these nutrients. These emerging fields could be improved to enhance personalized diet consumptions trends depending on individual genetic makeup and to develop innovative functional foods based on genetic pattern. So let's see the interactions of nutrients and the gene expression, that is nutrigenomics. Nutrients can act on our genes and can change the genetic expressions or phenotype by following pathways as shown in the diagram. Nutrients act as a ligand for transcription factor, receptor, and affect the normal cell growth. Also, diet influences gene expressions by metabolism. These influences can be done either primary or secondary pathways by altering concentrations of substrates or intermediates. Further, normal cell growth can be altered by diet consumptions proceeded in single transduction pathways as shown. Researchers have revealed the interrelations of live cells response to varying environments with modified gene expressions, and also they have studied how nutrition can affect the proliferation and differentiation of cells. Therefore, nutrigenomics studies on basic and applied nutrition in food research furnishes new insights into the influence to food ingredients such as carbohydrates, proteins, fats, carotenoids, vitamins, minerals, flavonoids, and edible phytochemicals at the molecular level. Further, nutrigenomic applied areas includes food safety, food authenticity, researches in genetically modified organisms, that is GMOs, and personal food planning. So let's see the flowchart. In this, first that we take the diet and the nutrients that is present in it, that are metabolized in our body and then they are interact with the gene or gene expression so that the cell growth takes place. These are the different pathways of how nutrients act on the gene expression in normal cells. Let's talk about how does nutrigenomic testing work. The DNA test for nutrigenomics is a simple swab of the inside of your mouth that's sent off to a lab for analysis, and you get the results a few weeks later. Labs may analyze 70 or more different genes. Nutrigenomic testing is based on science and is very accurate. So let's talk about the advantage of the nutrigenomics. The intention of both of these technologies are to achieve more effective individual dietary intervention strategies aimed at limiting disease, enhancing the quality of life, and managing healthy aging. Main advantages of these technologies include improvement of health and preventing diseases through tailored diet and lifestyle prescriptions, and effectively control chronic diseases. And there are some disadvantages also, which includes attention is drawn away from other modifiable risk factors, focus only specific nutrients or foods, misleading the claims, and increased cost associated with personalized diet and designer foods. So, what are the applications of the nutrigenomics? The applications of the nutritional genomics are multiple. With personalized assessment, some disorders like diabetes, metabolic syndrome can be identified. Nutrigenomics can help with personalized health and nutrition intake by assessing the individuals and make specific nutritional requirements. The focus is in the prevention and the correction of specific genetic disorders. And the examples of the genetic related disorders that improve with nutritional corrections are obesity, coronary heart disease, that is CHD, hypertension, diabetes mellitus type 1. Then genetic disorders that can often be prevented by proper nutritional intake of parents include spina bifida, alcoholism, and phenylketonuria. So let's talk about these diseases in detail. So first is a coronary heart disease. Genes tied to nutrition manifest themselves through the body's sensitivity to food. In studies about CHD, that is coronary heart disease, there is a relationship between the disease and the presence of two alleles found at E and B apolipoprotein loci. These loci differences result in individualized reactions to the consumptions of lipids. Some people experience increased weight gain and greater risk of coronary heart disease, whereas others with different loci do not. Research has shown a direct correlation between the decreased risk of coronary heart disease and the decreased consumptions of lipids across all populations. So the next disease is the obesity. Obesity is the one of the most widely studied topic in nutritional genomics, and these are the most common diseases in our daily life. That is occur due to the consumption of the fast food mainly. Due to the genetic variations among individuals, each person could respond to diet differently. By exploring the interaction between dietary pattern and genetic factors, the field aims to suggest dietary changes that could prevent or reduce obesities. There appear to be some SNPs that make it more likely that a person will gain weight from a high fat diet. For people with AA genotype in the FTO gene showed a higher BMI compared those with TT type genotype when having high fat or low carbohydrate dietary intake. And the next is the phenylketonuria. Phenylketonuria, otherwise known as PKU, is an uncommon autosomal recessive metabolic disorder that takes effect postpartum, but the debilitating symptoms can be reversed with nutritional interventions. And there are the future trends and the challenges that we have to face that are as follows. Nutrigenomics is the application of high throughput genomics technology to nutrition studies. If it is used correctly, it will promote a better understanding the effect of diet on homeostatic control and the metabolic pathway. The regulation is disrupted in the initial stages of a diseases related to diet. In the future, nutrigenomics will enable efficient dietary interventions to restore normal homeostasis as well as prohibit diet related diseases. New commercial representations of the individualized nutritional advising established on a person's DNA were also highlighted as a result of knowledge integrating technology into health sciences. In this approach, organizations concerning the healthcare system must act to control these commercial representations in order to protect patients' honesty while also improving the organization's nutrition focused performance. So, come to the conclusions. Nutrigenomics are emerging disciplines that are expanding a variety of fields of study with medicine, heredities, and diet. Population studies, large sample sizes with proper experimental design, scientific trials, and product specific trials in people selected for specific genetic variations must now be empowered and examined using the massive amount of data obtained by GWAS. The ultimate purpose is to make more effective dietary interventions for individual technique, for preventative medicine, and for the better quality of healthy life. Indeed, knowing very relevant to modulations in gene and diet could assist to profile the general dietary recommendations for each populations from a public health perspective. So these are the references from where I had collect the content, and the lastly, thank you.